Good evening and welcome to A Slice of Life. I'm your host, Jim Therian. Before you were born, actually long before you were born, there was a tree that dominated the eastern deciduous forests in and around the Appalachian mountain range, including Connecticut. It was described by many as the perfect tree. It had lumber, it had nuts, medicine, it had everything you would want. The best guess was back then, there were millions of these trees all along the Appalachian forest. Millions of them. But then something happened. Which brings us to the subject of tonight's show, our guests both in studio and Zoom. Both in studio and Zoom. I speak of the American chestnut tree. Now, if you've not heard of this tree, the American chestnut tree, well, there's a reason for that. We have with us two scientists from the Connecticut Agricultural Experiment Station over in New Haven. We have a Dr. Sandra Anagnostakis on Zoom, and in studio here we have Dr. Susanna Kerio. Mr. Jack Swett from the local chapter of the American Chestnut Foundation could not be here tonight as he is away on business. So welcome to the show. Dr. Sandra Anagnostakis, you're up first. Could you please tell us about the American chestnut tree?
Absolutely. I started working in Connecticut in 1966, and my initial work was on fungi that caused plant diseases. And then a colleague, Richard Janes, came into my office and said, "Here, you're a mycologist. Why don't you work on this?" And he gave me a branch of an American chestnut tree that had signs of a serious fungal infection. I looked at it and said, "Oh, everybody worked on that in the 20s and 30s. There's nothing more to be done." But I started looking more carefully, and I made some cultures, and I did a little work and found that, in fact, there was a lot more that could be done. And that was the beginning in about 1968 of my involvement with chestnut trees. Richard Janes was the geneticist who was breeding chestnut trees, following in the tradition of Arthur Graves, who had begun the work in 1930, working with Donald Jones at the experiment station. Richard Janes was a student of Graves and Jones and made many very important crosses of chestnut trees between Asian and American, and wrote a lot of papers about the resistance to chestnut blight and the ability of trees to become actual timber trees. Now, American chestnuts were timber trees. Yes, they made nuts, but the nuts were very small. They were good food for wildlife, and many people enjoyed them, mostly in the Appalachian Mountains in Tennessee and so forth in the South, where the trees were really a major part of the forest. Here in Connecticut, there certainly were chestnut trees, but many of them were planted hybrids of European and American, or even with Japanese and American. And the potential for those chestnut trees in Connecticut was mostly considered to be their ability to grow really tall. And as you can see in this picture, this is a tree from Connecticut, Scotland, Connecticut, in 1905. It was part of an experiment station study on the woodlot and the use of the trees in the woodlot for timber. This tree was cut, of course, in the process of this lumbering. It was measured to be 103 years old, and it was 83 feet tall. That's about the size of a Connecticut American chestnut tree. They got to be about 80 feet tall, and this one was 27 inches in diameter. It was therefore an important part of the lumber industry. Now, the American chestnut trees were assumed to be there forever. And so people were using them and cutting them down, and they sprouted again from the root base. So there was no problem. There were going to be more. We have many pictures from experiment station research showing sprouts of chestnuts that are 50 years old after having once been harvested and then sprouted. And those sprouts were grown and used again as timber. And they were used for lumber and telephone poles. And if you look at my webpage, which you can find on the Experiment Station website, you will see a page that describes some of the early use of chestnut in Connecticut, including my absolute favorite, which was in pipe organs, because there was a big industry of producing both pianos and pipe organs in Connecticut. The pianos, of course, came from Ivoryton, and they used chestnut for the bulk of the production of the pianos, and the pipe organs were mostly along the coast. So not only for timber and telephone poles and railroad ties, but also musical instruments.
Doctor, if I may interrupt a little bit. From the first photo that we saw, a staggeringly healthy looking tree, it's amazing to me that this tree a tree that size could be killed by something as simple as a fungus. It's just amazing to me.
Yes, the fungus came in on Japanese chestnuts that were first imported in 1875. And the fungus was present on Japanese chestnuts, but not causing serious disease. And these Japanese chestnuts were planted in many places in Connecticut, starting in Stamford, where they were first put in. And then, people said, these trees are different from American chestnuts. They produce nuts earlier. The nuts are much larger. And they were planted everywhere. And one of the things I have done is to go around Connecticut looking for some of those original 1876 trees. My absolute favorite is at the former Bee and Thistle Inn in Old Lyme, Connecticut. There's a lovely Japanese chestnut planted in 1876 from the first importation. It has chestnut blight all over it with large swollen cankers. The tree is totally fine. It is ignoring that infestation of chestnut blight and producing nuts every year with pollen from across the street, another importation of chestnut trees.
Now, I wonder if we could go to the third and fourth photo where it actually shows you in action. You're up a tree and it looks like you're doing something to the tree.
Yes, There I am, up in a tree. Those days are gone forever. We heard about a report from Italy. My colleague Richard Janes was keeping very close tack on the chestnut literature and found a report from Italy that chestnuts where there was chestnut blight were recovering from the infection. And the guy in Italy said, "Oh, well, we don't have to worry anymore. Those chestnut trees are recovering. They're going to be fine." And the impact of this on all of the chestnut industry was really important. And a scientist in France named Jean Grant went to Italy and took samples of those apparently recovering chestnut trees, brought them back to his lab, and found that, in fact, there was something wrong with the fungus. He didn't know what the problem was, so he worked on it, did a lot of research and some very important things showing that whatever was wrong with the fungus could be transferred if he put bits of this Italian fungus into a killing canker on a chestnut tree in France, and the tree would recover. So I wrote to him in 1968, and he sent me cultures of these unusual fungi. At the time, we were working on viruses in fungi in the genetics department at the experiment station. And very quickly, we showed that this was, in fact, a virus in the fungus. And we did a bunch of tests, and we were able to show that the virus was able to keep the fungus from killing trees. So we thought, wonderful, we're going to save the world, we're going to cure chestnut blight. And we began to make tests of these infected fungi. We call them hypovirulent because they're less than normal virulent on chestnut trees, but also they have this curing property. So we inoculated them into chestnut trees at the experiment station farm after we got permission from the USDA. And we found that we could keep chestnut trees alive by putting these hypovirulent fungi into the killing cankers on chestnut trees. So that picture of me in a tree is me inoculating the hypovirulent fungus into a killing canker on an American chestnut.
That's wonderful. We're going to have to go to break now. Dr. Sandy, I certainly thank you for your contribution tonight. We're going to watch a quick video, and then Dr. Kerio will be speaking. So once again, thank you for your contribution to this wonderful mission, I suppose. And we'll see you. We'll be in touch. Thank you.
You're welcome.
Hi, I'm Florian Karl. I'm the Secretary and Research Coordinator of the Connecticut chapter of the American Chestnut Foundation. I grew up in southern France, where chestnut is it's pretty much everywhere. We find them everywhere, we eat them for the holidays, and it's really an important flavor palette. And when I moved to the US in 2014, I couldn't find them anywhere, not at the store, not at the market, and I couldn't find any trees anywhere in the States. And luckily, I ran into Jack Swat, who's the president of the Connecticut chapter at the Durham Fair. And he was there to educate the public about the tragic loss of chestnuts in the US with the blight hitting the tree in 1904. And this is where I learned all the work that they are doing to restore the tree to the native range. And this is how I decided to be involved in the foundation. The American Chestnut Foundation was created in 1983, and 16 chapters in all the states on the East Coast that had chestnuts in the past are working together. We work with scientists, we have partnership with universities and research groups like the Connecticut Ag Station, where we are here. And the idea is that how can we use all the scientific knowledge and tools that we have to bring back that tree? We work on the mission of given by the scientists at the headquarters in Asheville. And this is where we try to work on local concern for Connecticut. So for example, we try to find wild trees that are still producing nuts or who are just isolated enough, and then we cross them with pollen from other trees to try to harvest nuts and save these. And so we save the genetic material of these trees into orchards. So we have about 15 orchards all over the state that contains material that is specific to Connecticut. So we try to save the diversity of the Connecticut trees to bring them back once we are able to cross them with the trees that are resistant to the blight. One of the success we're very proud of is we are finding more and more wild American chestnut trees. In 2021, we found more than 500 new trees who can potentially be new source of genetic materials. With our help and the help of the volunteers in our chapter, we have some of the nuts produced by this tree are saved in our orchards. And the fact that we are able to use lands that they generously lended to us through either land conservation and land trust or by private citizens who give us access to our land, I think this is really the most successful thing that we can do in our chapter, like seeing everybody under the same cause trying to put everything they can to bring that tree back. The future is very bright for the American chestnut. We are really at the cusp of a scientific breakthrough with Darling 58, which is a new tree that is blight resistant. Using the enzyme of wheat, it's naturally protected against the blight and it's blight resistant. By crossing this tree with the trees that we have in our Connecticut orchards, we should be able to bring back trees that are resistant to the blight, but also resistant to the Connecticut climate. Bringing the chestnut tree will not only help us to restore the forest and the mistake we've done in the past, but also will show us how we can do that for other species and help us combat different diseases. There's two things really challenging about bringing back a tree that is functionally extinct. One of the challenges, we don't have a road map to know how to do and how to bring back a species that is functionally extinct. The other challenge is as the population age, the people who knew the tree, who experienced the tree when they were younger, are less and less representative in the population, and the younger people do not really know what a chestnut tree is, why a younger population should care about the chestnut. And so that's why we're doing a lot of outreach events to try to educate of the importance of the tree. Everybody can join the American Chestnut Foundation. This is a very open organization. The idea is that, of course, you will have scientists working in the foundation and in our chapters, but everybody who's interested and wants to help in a mission is very welcome to become a member. You can contribute as much or as little as you want. For example, if you're just interested in learning more, you can come in hikes with us, but you're also very welcome to volunteers and help us plant nuts in our orchards, maintain the orchards, go on fair and explain to the public why we care. So it's really whoever wants, and we're very happy to include as many people as we can.
Welcome back. That was from the Connecticut branch of the American Chestnut Foundation. Now with me is Dr. Susanna Kerio. She also works at the Connecticut Agricultural Research Station. And Dr. Kerio, please tell us what your story is.
Well, thank you for having me on the show, Jim. So I want to start by telling the connection that I have to the chestnut blight. So my background is in forest ecology and forest pathology. I studied at the University of Helsinki in Finland. And during the very first forest pathology class that I had in school, I learned about chestnut blight. And you know, forest pathologists and plant pathologists, they have this kind of weird fascination with diseases. It's like, oh, cool, what is this fungus or whatever, you know, bacterium is doing to the tree, and wow, it causes death. But then I never had seen chestnut blight in the wild before I came to the US. So I actually, when I started at the Connecticut Agricultural Experiment Station in 2020, one of the first things that I did, I got in touch with Dr. Anagnostakis, because she had this knowledge of the chestnut experiments that have been established in Connecticut over the years. So what I did, I went to one of her old field plots in Vernon, Connecticut, and what I saw there was just devastation. Dr. Anagnostakis had planted about 100 chestnut trees 10 years ago in the field plot, and only a handful of these trees were surviving.
Wow.
That's when I understood that this is a really serious problem caused by the chestnut blight for the survival of the chestnut tree.
Right.
And that's when I got very interested in contributing to the research that's aiming at restoring the chestnut tree as part of the hardwood forests in eastern United States.
Now, you have some display items here. These looks like, I wouldn't say petri dishes, but they look like growing mediums of some sort.
Right. So these are tissue culture vessels. So these relate to the research that I'm conducting currently at the Connecticut Agricultural Experiment Station. So this relates to developing methods to clonally propagate the chestnut tree, because chestnut tree it is not really possible to propagate these chestnut trees from rooted cuttings. The success rate for this method of propagation has been very low. So in this sense, we need alternative methods to be able to produce clonal material. So why would we want to produce clonal material trees that are genetically identical? Well, for instance, in the case of chestnut blight, if we are able to find a chestnut genotype that has the high blight resistance, then we might be interested in propagating this tree and cloning it so that we can establish this tree in several different environmental conditions, and then improve the stability of the ecosystem where chestnut used to be an important part.
Now, these things in this growing medium, are those clones?
Yeah, so these are actually somatic embryos. Somatic embryos are embryos or sort of like seeds that have been produced from any type of normal plant tissue. So how we induce the production of these embryos is through growing these, the plant tissue on this tissue culture media, either with plant growth regulators or without them. And then this way we are able to induce them to convert into entire plants that have both roots and shoots. And this way we can produce hundreds of genetically identical plants.
And that would be just for like experimenting to see which one rises or beats the fungus?
Right, exactly. And then this also gives us the opportunity to, when we have the plant material that's genetically identical, it's possible for us to conduct these various experiments and challenge the trees, for instance, with different strains of the fungus, or expose them to drought or some other stress factors such as insects. And this way we are able to identify the genetic components or genomic regions that are associated with resistance to blight, drought resistance, or pest resistance. So having these clonal reproduction methods is a component in the breeding program, or the efforts to breed a blight-resistant tree.
Wow, that's like heavy-duty science stuff there.
Right.
Cloning and all that.
Right.
That's amazing.
Yeah. But I just must say that none of this would be possible without my amazing collaborators. So I would say that this is a huge challenge, restoring a tree species back to the nature. So no one research group would be able to do this alone.
Wow.
So for this, we need like a network of collaborators from different research institutes, different universities, and research stations.
So this is all wonderful work that you and Dr. Sandy are doing, and do we have a timeline when I can go down to the local nursery and pick myself up an American chestnut tree?
Well, that might already be possible nowadays, but the chestnut trees that you would be picking up from the nurseries would not have very high blight resistance. Yeah, for instance, the Connecticut Agricultural Experiment Station, we have a very diverse collection of different chestnut trees and different species of chestnuts. So those seeds and nuts, they are available to nurseries and citizens who are interested in growing their own trees.
Well, go ahead.
Yeah, but I must say that there is some hope in getting resistant trees to the market, possibly within the next decade. This all depends on whether the deregulation of the Darling 58, which is a transgenic American chestnut tree, will be passed by the federal agencies that are now considering the deregulation of the transgenic
And on
tree.
that note, Dr. Susanna, we have to call it a day.
Okay.
Thank you so much for your contribution, and sadly, we're out of time. It's just wonderful work that you're doing to help restore this magnificent tree, and we are certainly very thankful. So that's it for a Slice of Life, American chestnut tree. What a wonderful, wonderfully complex endeavor you're on. Thank you and good night.